Silvina is an assistant professor and a small fruit extension specialist, and she's going to present the blueberry horticulture update. Please welcome to Dr. Rubio.
Hello, everyone. Thank you for being here this afternoon. Once again, my name is Silvina Rubio, and I'm the small fruit extension specialist for the state of Georgia, and I've been with the University of Georgia since 2021, and I am located in the Tifton campus. Today, I'm going to talk to you about 3 topics. We're going to talk about fertilization and pruning and ethaphone applications to delay bloom. So, but also before I start speaking about fertilization and these other topics, I would like you guys to help us by scanning the QR code and answering some questions about fertilization. It's been several years since the fertilization guidelines for the state of Georgia had been established, and we're trying to redesign the fertilization guidelines and the sufficient levels for blueberries grown in Georgia. And the reason for this, it's because we set up these fertilization guidelines and these sufficient levels based on older varieties that are no longer planted. So now let's move on with fertilization. So there are key questions here to ask. Probably many of you are producers or are interested in growing blueberries. And the questions here are like, how much is needed? How much actually the plant needs, or how much the soil needs for the plant to have the adequate yields and fruit quality. It is also important to know what we have in the soil already. So doing soil samples before establishing a blueberry field, and to know the pH of your location and what the amount of nutrients that the soil has, it's very important. Then through the production cycles in your blueberry production system, it is also very important to take leaf samples to know, when the plants, what the plant sufficient levels are and what is the nutrient content in the plant. But it is also very important to know when this plant needs that fertilizer. Probably a lot of you are not fertilizing at the moment because it's winter time and it's not, those roots are, are dormant. Also, it is very important to take those leaves tissue samples after harvest, and that is what we usually recommend for Georgia growers. So also it's very important to know how is the fertilizer taken up by the plant, the nutrient mobility of each of the nutrients that we apply to the soil and are taken by the plant. So here it's a table in which tells you what is the nutrient mobility. We all know that nitrogen is very mobile in the plant, and also phosphorus is mobile in the plant. But if we look at calcium, we see that calcium has very low mobility in the plant. So several times we take those leaf samples, the leaf tissue samples, and the calcium levels on those leaves are not equally or to the calcium levels in the fruit. And that is because calcium has a very low mobility and does not move from the leaves to the fruit, sometimes when the fruit needs it. However, if we look at the soil, for example, if you look at the table over there, you can see the nutrient mobility in the soil. We are very familiar on the sandy soils in Georgia and also in Florida, the nitrogen leaches because it moves really fast. So phosphorus has also low mobility, and calcium has also low mobility. So if calcium has low mobility, we recommend our calcium application, via a foliar application. Well, this is still something that is not, there is a lot of data from the Pacific Northwest looking at calcium foliar applications, and they did not find any correlations between foliar calcium applications and increase in calcium in the fruit, or reducing fruit quality, or increasing yield, or fruit firmness. So in the state of Georgia, this extension publication was developed by 2019. This is by my predecessor, and this is how much we recommend for nitrogen fertilization when the plants are established. So if you want to, have this full extension publication, I'll recommend visit the link that is on that below that table. But still, a lot of these recommendations were based on a lot of varieties that are no longer planted. So we also need to look at do we actually need this amount at that time. This is for the second and third year. Again, you can get this publication online, and you will have the full publication. This is only a screenshot of the publication. So that is how much we recommend for the second and third year, about 90 pounds of nitrogen. And we recommend those between 4 to 6 applications, depending if we have the granular fertilization or fertigation, about 3.3 pounds of nitrogen weekly. For phosphorus, we have that recommendation there. If the soil is less than 20 pounds per acre, we need to incorporate 240 and 300 pounds of phosphate to increase 10 pounds of phosphorus in the soil. And the potassium, some lower levels for potassium in the soil is like less than 100 parts per million. I'm sorry, in the leaves. So, but once again, all of these guidelines, all of these fertilization guidelines, and all these recommendations were, basically established when the industry in Georgia started. And these are the sufficient levels here, and you see they date back from 1999. So it is time for us to look again one more time and, and do studies and research on the new varieties that are planted in Georgia. In Georgia, one of the largest planted variety is Fardin. That is a variety that came from the University of Florida. And a rabbit-eye variety that is widely planted, it's Brywell. So when I took the position, I started looking at data, looking at the updated research on blueberry fertilization, and a lot of these were developed in the Pacific Northwest. A lot of research on fertilization was done at the Pacific Northwest, and it was done by Bernadine Strick. So she showed this graph one time in which, when we were asking that question, how much is needed, how much of these nutrients are in the soil, how much nutrients are in the plant, what do we need to apply, when do we need to apply. We also need to look, at the blueberry production system as like things go in and things go out, and one of them are the nutrients, right? Because when we harvest fruit, we remove nutrients out of the plant. That's what we have to incorporate nutrients. So looking at this graph, I found that phosphorus is actually taken out by the system in like really minor quantities compared to other nutrients. So when we harvest the fruit, that fruit has very little phosphorus content. So I wanted to kind of like validate that and see if we will find the same thing for southern highbush or rabbit-eye, because this data is, and these studies were done on northern highbush. So I did the fruit analysis, and I found similar results in which we are taking phosphorus. When we harvest the fruit, we are taking very minimal phosphorus out of the system, and the fruit has lower phosphorus content compared to potassium. So that led me to start some kind of like surveying and taking soil samples and leaf tissue samples from different farms in the state, and with different management practices, trying to like coming up with like redesigning fertilization blueberry practices for the state. So in this kind of like survey of collecting leaf and tissue samples and seeing how much variation we have with different management practices in each of these different farms, I set up kind of like three farms in which I took leaf tissue sample every 2 weeks. So we have a farm right there in Nehana with having the premier blueberry. Let's see if I can get this to work. Okay, so we have Nehana, so three locations, Nehana, Alma, and Hoboken. The Nehana had Premier that was established in 2009, and the Alma had Fardin in 2019, and then the Vernon. These were the same, kind of like the same management, but at different locations within the farm. And then I had the Hoboken farm that had farding, which was the same farm that variety that we had in Alma. And they were, had different spacing, and then they also have different fertilization practices, as you can see here. And the current fertilization practices per plant were also different. So I wanted to see if there is any variation, if there is something that we can find out is like if we have lower fertilization, are we actually seeing that lower, nutrient content on the leaf tissue, and trying to see if we can find anything that needs to be changed or that lead us to any changes of fertilization. So this graph here, I'm going to show you. It's this is from the Nehana farm in which we sampled Premier, and if you can see right here, we measure the soil at different depths, and we had young tissue and old tissue were also sampled, and here in that this green line, it is the recommended level in the soil. And this is the recommended level on the leaf. This is the sufficient level. So if we're below this, we can say that we need to incorporate phosphorus. As you can see there, with those fertilization practices that they had, we had that the recommended levels of phosphorus in the soil were above the recommended levels, and then the ones in the leaves were also above the recommended levels for Premier in Nehana. Not advancing. And then when we look at the Alma, the Vernon, we found that the phosphorus levels in the soil were right there, where we can say that we need to incorporate the phosphorus. And then here we can see that actually the phosphorus levels in the leaf tissue were above the recommended level. So there was not actually correlation. We did not see low phosphorus in the soil and low phosphorus in the plant tissue. For that, also on the southern high bush farding and the same farm in Alma, we see how phosphorus in the soil, it's above the recommended level. However, we see some lower phosphorus content in the leaves. So we did not see like a correlation. We did not see an effect on the soil and in the leaf tissue. And that Hoboken farm was very surprising for us to find this data because the phosphorus levels in the soil were really low, and they were below the recommended level. However, we did not see a higher impact on the leaf tissue. There were, some of the results that we got were above the recommended level on the young tissue. So if you can remember from this table, like this is that pounds per acre each grower was reporting, and actually the Hoboken farm had higher pounds per acre. So we did not see any correlation between what it was the amount of phosphorus that was found in the soil and the amount of phosphorus in the leaves. So this is something that we're going to continue looking at. We're going to study phosphorus because phosphorus also affects the mycorrhiza that are in blueberry roots. And we're probably going to establish some experimental trials in which we deplete that phosphorus or we don't incorporate phosphorus in the soil. And if you were here yesterday, you probably heard Gerardo Núñez from the University of Florida talk about his work on phosphorus. And him and I are working together in trying to elucidate a little bit more what's going on with phosphorus and the physiology of phosphorus in blueberry plants. And if you want more information, this is a publication that just came out from my lab, and you can access it online if you just type assessing phosphorus fertilization in blueberry production. So now moving on to fall pruning. I know this is not a practice that we usually do in Georgia, but I come from a peach background. I did a lot of work in perennial fruit crops. I work with avocados, mangoes, and table grapes and peaches. So when I moved to and I took over the position, I was surprised that we don't do any fall pruning. So I decided to look into it. I also look at, in the literature about fall pruning and what pruning does to blueberries and how can it be incorporated. And I know this practice is done in the Pacific Northwest and I think also in New Jersey. So I wanted to see what happened. And here you can see, a plant that was pruned right here compared to a plant that it's not, pruned. So we have a more open canopy in the prune plant. We kind of reduce the crop load, and crop load, it's being, heavy crop load has been linked to deficiencies in calcium and boron. So the more we hedge and the more we increase, the vegetative growth, we can start reducing calcium and boron in the plant, and also that. The magnesium and calcium ratio that also it's very important for fruit quality. Here are more pictures of that of the experimental trial that I said these experimental trials in 3 farms, one in Alma, one in Pearson, and one in Hoboken. So here are the treatments that we established. There were 6 treatments, and depending on, on the farm, because with some growers, sometimes it's like, hard to coordinate when we can go and prune or set up this experiment. Some of them I do have, like at the Alma farm, I did establish the 6 treatments, the 6 pruning treatments, but in Hoboken there is only 5, and for Pearson there is only 3 treatments. So we had a hedge summer after harvest, so that meaning the common commercial practices that we do after we after the harvest is over, we come and hedge the plant, and then I went and hand pruned in November. That treatment that is on red right there, it's the commercial practice, so that's the control, and this is where we hedge, and then you guys tip in the fall. And then the 3rd treatment, it's not hedge after harvest, so we didn't do that, the regular hedging, and then I went and hand pruned. And then I hand pruned during the summer, and then I hand pruned during the fall. So this hand pruning summer was done right after harvest. And then we have a control treatment in which we didn't do anything. And then I added one more treatment here is that we hedge. I asked the grower to hedge, and then right after he hedged, I came and hand prune, and then I hand prune in fall. So now I'm going to show you some results for this experiment. So what we were expecting here, we expected that we will decrease yield when we hand prune, because that was the expectation, that we will have lower yield probably on the plants that we prune, and we will have a higher berry size. So that was kind of like something that we were hoping to see. But as you can see here, this is the one that is in the red oval. This is the commercial practice. So as you can see here in Alma, we did not find any significant difference between that commercial practice or that hand prune in summer on the fall. We found some significantly difference with the hedge in summer and hand prune in the fall, and that hedge summer, hand prune, summer and fall. And then there were no significantly difference with the hand prune on the fall either. For the Hoboken, we did not find any significant differences. And in the Hoboken farm, we had a little bit of issues with keeping the treatments truly with not hedging at all. So that might be affected our results. But also, we did not find significantly difference between the commercial practice and hand pruning in the fall in Pearson, as you can see here. So also we saw some increase in berry weight, which I mentioned that was kind of expected. So you can see how the commercial practice had a lower berry weight compared to the hand prune here. And that was seen also in Pearson once again, but we did not see any differences in Hoboken. So when we look at berry size, we saw something that, also that it was expected, that we had higher berry size in the ones that were hand pruned compared to the ones that it's the commercial practice. But we also saw that in Hoboken, in which we didn't see, the yield differences. And we also saw that in Pearson. But in here, we did not see that significantly differences with hand pruning. So here, when we were analyzing the data, and for example, the Hoboken farm, we established it. We did the hand pruning later compared to like the Alma farm and the Hoboken farm. So the Alma farm is kind of like that we felt that it had the more sound results just because the way the field was managed and the experiment was managed. And that's what we put together all this data here with all the pruning treatments. And you can see it's the yield per plant right here on the bars. And the dots are the berry size. And the color symbolize the berry weight. Okay, so we are in the berry weight around these, but look at our dot line, and that's the berry size that is in this axis. So we are a little bit above 15 millimeters on berry size. So what does pruning, the potential for this is that pruning, yes, it's definitely a cost for the growers. If we hedge and prune, it will be increasing production costs. But if we move only to doing the fall pruning, we can also obtain, higher berry size, which it will pay about 20% higher the price compared, to that to our regular or smaller size. So we also have to add a little bit more cost to it. I've been timing myself every time I prune so we can put a number to this and see how effective it will be. And we all know also that the market is moving to a larger jumbo berry. That's what the consumer it's been preferring. So there might be potential for this. Also, what we did is that we looked at some developmental stages and how pruning affects the developmental stages. And that is also something that it was very, very important for pruning, is if we do that, we compare that hand pruning in the fall to the commercial practice right here, we have more fruit coloring in the hand prune compared to that commercial practice. And one thing that pruning does is that it concentrates ripening, and that will allow you to harvest the fruit probably 2 or 3 times. And if we incorporate pruning, we also have to think that it's going to increase machine harvest efficiency. And this is something, again, that was proven in the Pacific Northwest. And we can also concentrate ripening. So if we machine harvest, it will be probably reducing our green fruit drops and a loss of fruit in the production system. I'm just going to run into the fruit quality things. I think I'm running out of time here. So we did not see significantly difference in total soluble solids, but we saw an increase in firmness of the berry when we hand prune compared to the commercial practices that are here. So also, just to finish up with my talk, I know there's a lot of growers that have been trying Ethaphone applications to delay bloom, and I know this has been done before by Gerard Kruger, and in rabbit eye at least delayed the blooming by 7 to 10 days. And as I said, Ethaphone has been applied by growers, and but there is still a lot to know about Ethaphone on when is the best time of application, the effect of temperature on the effectiveness of Ethaphone, and what are the mechanisms actually the Ethaphone uses to delay bloom. And this is specific for bloom. I know some other presenters talk about plant growth regulators and how Ethaphone will enhance ripening and all, but this is specific to delay bloom, and of course avoid the spring freezes. So we had 4 treatments here, 200, 400, and 800 parts per million, and I'm gonna just run really fast because I'm running out of time. This is a variety farthing. We found significantly differences here at the ripening stages and at bloom a little bit, but see, it was more detrimental to that. It was meaningful for the ripening stages here. We also found significantly difference for Georgia Dawn, in which it delayed the bloom of Georgia Dawn, and I'll give you more detail on the following slides. So here you have, there were significantly differences on also the ripening of the berry. And for kikris, we found significantly differences, as you can see above, in different sampling days. And we went and took some bloom counts and count the bloom to see when was the plant blooming, if it actually had a higher percentage of bloom flowers that were blooming, or flower buds that were blooming. And this is kikris also, like some differences on ripening, because ethaphone could not only delay blooms, but it can also delay the ripening. So there is by about a week, that's what is being reported. So for farting, as you can see, S8, it's the blooming stage. We did not see any significant differences here, but we saw it on fluid coloring here, in which the control had a lower amount of fruit that was coloring. So that's kind of like something that it goes, it's different, it's contrary to what is being reported on rabbit eye varieties, but this is what we obtained for farthing. For Georgia Dawn is our early cultivar, and we saw some significant differences on blooming. As you can see here, the control had a higher amount of flowers that were blooming compared to the other treatments, and we found that this was the higher dose of ethaphone had lower amount of blooms. But it also delayed some of the, but it also had effects on the ripening. As you can see here, the control had higher fruit coloring compared to the higher dose, and the 200 parts per million. So that we're seeing some effect also on fruit ripening for Georgia Dawn. And here on the kikris, where we found significantly differences, we found also like on S6 that we had that the control had high amount of flowers blooming compared to the higher treatment of ethaphone. So here you can see this is at so here you can see it better, how you can have more blooming in the control compared to the treated plants. So some general remarks. So reducing phosphorus fertilization could be a possibility to reduce fertilizer costs. We still have to do more research on this, but it seems like that might be the way that's something that we might be recommending in the future. Pruning increased berry size without affecting yield, which can lead to premium prices for growers. And ethaphone is very effective on delayed blooming in Georgia Dawn, however, not for farthing and kikris. It's still, we need to validate this by doing a second-year experiment. And these are more topics of research that we're working on. Calcium and boron is very important for fruit quality. And black bud, that is an issue that growers have been reporting on farthing. Sap analysis nutrients, and looking at nutrients deficiency, aluminum toxicity, and maybe new amendments. And with that, I don't think I have time for questions, but if there is any, I'll be happy to take them. No questions? Okay. Thank you.
Thank you so much, Dr. Silvina Rubio, for this update in the blueberry field. Our next speaker is Dr. Rachel Idle. She's going to talk about the new freeze protection strategies for Georgia blueberry production. Please, welcome.
All right. Well, good afternoon, everyone. Thank you for sticking around this afternoon to join us for this talk. I'm going to update. I had a talk last year on nanocellulose, so this year is an update of what we've done and what we're currently seeing. So I'll save some time and go through this fairly quickly. I think everybody in here is probably in blueberries, but in case you're not, blueberries are a huge component of our state's agriculture, and Georgia is a big boy in production for the U.S. To put it in perspective, in 2022, we had a freeze that wiped out about half of the crop, and we still ranked 4th in U.S. value and utilized production, and 5th and is the largest producer. So Georgia produces quite a lot of blueberries. We produce primarily southern highbush and rabbit eye blueberries, and a common problem for both of them is a susceptibility to late spring freezes. They have the ability to just obliterate our crop completely. So most recently, we've had some devastating freezes occur. They occur every few years, unfortunately. In 2017, we had approximately 80% of our state's acreage lost to a late spring freeze event. And in 2022, we had 2 days of very hard freezes down to 25 Fahrenheit for about 50% crop loss. And you can see, let's see if this is my pointer. Yep. As we go from least developed or most dormant, so they're the most hardy for temperatures that are cold, to the most developed, which are the least hardy, so they're really susceptible to spring freezes. And oftentimes, the blooms are about in this stage by the time we get hit in March, if not more progressed. So current freeze protection strategies for blueberry in our state, and basically really anyone who's trying to freeze protect, are twofold. The first are overhead irrigation risers. The basic premise of this is we have sprinklers that are above the canopy system of the plants, and right before it starts freezing temperatures, you go ahead and turn on your system, and the water turns, it starts to freeze. So it goes from liquid water to solid water, so water to ice, and that change of state releases a few degrees of heat. So you run that your entire evening that it's freezing, and it continually changes from water to ice, and it releases a few degrees of heat, which protect the flower buds and gives them a nice giant coating of ice. Downsides to this are it can be very costly. If you have high wind conditions, it won't work. If it's too low of a temperature, it won't work. If your pipes don't go long enough down into the aquifer, it won't work, and so forth. So, or you don't have the right pressure. So there are limitations to it as well. The second most common method are wind machines. They basically serve to mix the air, so it works with a temperature inversion, also known as a radiated frost, where we have the warm air above the plant and the cold air down where the plants are growing. So it mixes it up to basically bring the warm air back down to prevent the freeze. The main drawback to this is that it's only effective with a radiated frost. So the bottom line is that our current methods have some limitations. They don't protect to very low or very low temperatures, so a new method is needed to at least add to our arsenal for freeze protection methods. So nanocellulose is a term that you've probably heard. I've seen it in Fruit Growers News and other things, in the Packer and so forth, talking about some nanocellulose work done out of Washington State. So what is nanocellulose? Very simply, cellulose is the material that plants are made of. So every single plant has cellulose. It's a polymer that's abundant in plants. In humans, we can't digest it, so when we eat salad, that helps our digestion. So basically, just the plant material itself is cellulose. It can come from wood pulp, plant fibers, any plant product. And it can be broken down into a very small scale, hence the word nano. So basically, nanocellulose is just very small plant products in cellulose form. And it can be divided or classified into fibrils or crystals. So if you go through a chemical means of making it, you get nanocellulose crystals, or if you mechanically break it down, it's fibrils. And it's often used in many other industries than agriculture's, for example, food packaging, foodstuffs, cosmetics, textiles, and so forth. And because of its properties, we're very interested in it for freeze protection. So it's known to be a good insulator. It's sustainable, biodegradable, and certified organic because it is just ground up plant parts, to think of it very simply. And in 2 published studies recently, it's been shown to improve heat retention by a few degrees around flower buds in some crops, and I'll go over that next. So using nanocellulose as freeze protection, it's currently being evaluated by Washington State, and they've published 2 papers on it. The first was in 2018 on sweet cherry and grape, and the results found that they improved cold hardiness by 2 to 4 degrees. So by spraying this on the plants, they improved the cold protection. Most recently in 2022, they also shared some results on apple and sweet cherry, and they found improvement of up to 5.5 degrees protection in both crops. And that is like double basically what we're currently able to protect with overhead irrigation and wind machines. And that's provided the right settings, the right environment for it. And they also showed that their cold hardiness continued to be hardy 3 days after application. So this might work for blueberry and other perennial crops too, but right now there's nothing known other than these two studies. So my program is the first to look at blueberry on this scale, and we've done so for the last 3 years. So our main project objective is look and see, would this work? Could we use it as freeze protection? Evaluate different compounds, concentrations, application methods to see, could this start to be a viable option to protect our crop? So this is a very busy table, but I want to go through some very simple take-home points. So in case you want all the information of the compounds is there, but let's go ahead and just not focus on that for a second and focus on the main parts, which are, we've done it so far for 3 seasons. In season 1, we focused on one compound in different concentrations. And the main reason why is when we opened this up, it's very thick. It's like a thick mayonnaise or a thick hand cream or face cream. And it's applied as a spray in the field, so it's going to go through your blast sprayer. And that is not going to go through your nozzles. It's going to clog it up and cause all kinds of problems. So we're like, well, we can't spray at that concentration. Let's give some dilutions a try. And you can see how chunky it is here. So we gave a few dilutions a try. The second year, we went with more compounds and one concentration. So we dialogued with the companies to try to get an optimal concentration to start with. Again, it's not currently used for freeze protection, so we were kind of trying to figure out what would be a good starting point. And in the third year, we again evaluated more compounds, but we also looked at application methods. So currently, it would be going through a blast sprayer, so we used a CO2 sprayer to mirror that. And then also we incorporated an electrostatic sprayer, which basically does just what it says. It has a static charge, so it makes it adhere to the plant more than a regular CO2 sprayer. And all 3 seasons, we evaluated STAR weekly. We did 5 weekly collections throughout the bloom season. And then in the third year, we had the unique opportunity, because we did not have a killing freeze, that we could actually evaluate a rabbit eye cultivar. So we chose Spritewell because it's our most widely planted rabbit eye as well. Okay, so the general preparation of this, we had a completely randomized design. And these were excised from the UGA Blueberry Research Farm in Alma. So Renee Holland and then Zach Williams and Keith Thompson worked their tails off to help get us the stems that we needed, and then would ship them overnight. And we would receive them in these little bundles. We'd stick them in little styrofoam trays and then spray them with the nanocellulose applications. This slide gives you a little bit of an example of what that looks like. You can see it kind of, these are dried nanocellulose. It kind of comes off in like a papery, like a sheet almost when they're dry. And the sample preparations, we divided them, the blueberry stems, into these small little chunks here, little 5-centimeter sections, and made these like little bouquets, almost like for a little doll baby. And we put them in our put them in bags and put them in our freezing chamber, and then did gradual reduction of temperatures, which is, these are just temperatures that are established by freezing tolerance experiments, so you can see if something's having an effect on freezing tolerance or not. We then, just like in the field, you can't always see if something got damaged right away, so we went ahead and put them in the refrigerator for a week and let them see, did they get brown or did they stay green, to see what was dead and what was alive. We evaluated those for 2 weeks afterwards, and we evaluated the top 3 stems for each of these. So basically, my graduate student on this was hopping for 2 years on this. She evaluated 120 buds per temperature treatment per compound. So it was a lot of work. And the way that we did it was basically under a dissecting microscope, so we could see them a little bit bigger. You see up here the standard that you're all familiar with, our bud development scale from stage 1 or tight bud all the way up to stage 7, full bloom and petal fall. And you can see here for each one of these stages, we would score them. We slice them longitudinally, so slice them in half. And if they were green, then we scored them as alive, because that means if they were in the field attached to the plant, they would continue to grow and develop normally. If they had any traces of brown, be it all the way brown or a portion of the style or stigma or stamens or developing ovary or what have you, anything that was brown, that fruit would either not develop at all or not to marketable qualities, so we classified that as dead. So the basic premise here is we wanted to see, we calculated how much was still alive, then we wanted to compare the treatments to the control group. Did it provide more freeze protection? So this slide, I'm just going to give a very quick summary of where we were last year, and then we'll go into our new results. So our very first year, again, this is all STAR that we did until the last year. So on STAR, we had 1 compound and 3 different concentrations. Now these are small, but this is just for the take-home points of this. And we evaluate them at 3 different temperatures. So LT10 means at this temperature, 10% of the flower buds were killed. At the LT10, we found that treated buds, so any treatments, some of them were significantly more cold hardy than the untreated control. So the way to look at these graphs is along the x-axis here we have collections, so collection 1, 2, 3, 4, and 5. At the earlier collections, you're going to see more of the bud swell, bud break, and then as we get into March, obviously we're going into petal fall and green fruit. Along the y-axis here, as it goes up, you see it goes from 2 or 0, -2, -4, and so forth. So the higher up it goes, the more cold hardy it was. So, and on the far right are the untreated controls, so it was just treated with water. So for example, these treatments showed more cold hardiness than the untreated control. When we went to LT50, the temperature that killed 50% of the flower buds, no differences were observed. And the temperature that killed 90% of the flower buds, we did see the opposite of what we wanted, in the sense of we actually had the control was more cold hardy than the treatment. So we're like, there's something going on here, but we can't quite see it. Perhaps these are just too dilute that we decided these concentrations to have the maximum effect, but it's suggesting that there might be something that protects at these early stages here. On to our second year, so this is where we're evaluating 6 compounds. The temperature that killed 10% of the buds showed no differences between the treatments, and the untreated control this year is this orange bar here. The temperature that killed 50% of the floral buds, we did see that the treated buds were more cold hardy than our untreated control here in the early stages. And again, suggesting that perhaps in those early stages, this is when this compound is working. The temperature that killed 90% of the flower buds, we saw 3 treatments providing some cold hardiness against the temperatures compared to the untreated control. And also 1 compound we saw had effectiveness in the late stages as well. And that was Cellulose. And we're still seeing in the middle parts of this, so this was like bud break, into the more progressed stages, the untreated control was more cold hardy than the treatment. So again, we're still not quite sure what's going on with this. Oh, the Cellulose providing protection in early and late stages here. Okay, moving on to the last year. For the 3 compounds, the year 3 for STAR, so this is with 5 compounds, and now we're looking at the CO2 sprayer, which mirrors a blast sprayer, an electrostatic sprayer that gives it a charge. We did see one of the Washington State compounds, it's WSUL, provided some cold hardiness in earlier stages, but then again we see, once it starts to get to the middle, we see the control group is more cold hardy. So something is happening there that we have to figure out. At the temperature that killed the 50% of the buds, we did see some promise again with the WSUL compound providing protection, and another Washington State compound number 47 providing protection in the late stages. The temperature that killed 90% of the buds, we did not see anything in the earlier stages. We did see again the Celluforce providing some protection in the late stages, as we saw in some of the other graphs, and another Washington State compound in the later stages. Now on to the final results for Brightwell. This is for 5 compounds, again, the 2 application methods. This slide, I think, is the most interesting out of all of it. We found 4 compounds that provided some protection against the freezing temperature. So one compound, Washington State C, was provided protection. Again, this is your control group here on the far right, so it's more cold hardy than that in the early stages. Washington State's 47 provided protection in early stages and again in the late stages, which is very promising. And 2 more compounds in Celluforce again for the later developmental stages, and another Washington State compound L for the later developmental stages. And interestingly enough, all of these were with the CO2 sprayer, so we're still figuring it out, but this is suggesting so far that maybe we don't have to purchase an extra piece of equipment and we can use the blast sprayer that you guys already have. At the temperatures that killed LT 50 or 50% of the buds, no significant differences observed, but there were the same trends that observed for the LT10. So again, you saw the Washington State 45 providing protection in the early stages and in the late stages, and Washington State L providing it also in the later stages there. For the temperatures that killed 90% of the flower buds, no significant differences, but the trends were similar. So this is interesting to me that we're still seeing the same compounds that we've been talking about, not significant, but the trends are there. For WSU 47 and WSUL. Okay, so that was a lot of information, so let me summarize the highlights. Overall, we're seeing some effect. We're seeing that likely nanocellulose is providing cold hardiness in the earlier stages of development and the late stages of petal fall and early green fruit. What we're thinking is happening is that the nanocellulose is providing more effect when the surfaces are smoother. So if you think of your flower buds before they develop, they are nice and closed before they start to open and have those beautiful blossoms come out. And then once the blossoms drop and petal fall, you just have a small green fruit. So it looks like it's adhering and protecting at those 2 extremes, but it's the middle part that we need to figure out. The 3 compounds that show currently the most potential are WSUL for early stages, WSU 47 for early and late stages, and Celluforce for late stages. But overall, these results are not consistent enough for me if you gave me a call and said, hey, Rachel, what should I spray and when and what concentration to protect my crop? I can't give you that quite yet, but we're making progress on it. And the fact that we've narrowed it down to 3 compounds to continue to study, to look at number of applications, increased concentrations, longer drying time, and perhaps coupling it with a protective coating. In my talk later this afternoon, I'll talk about a protective coating for fruit cracking, something that's more hydrophobic that might help as well. So with that, I'd like to thank our funding sources, the Georgia Blueberry Commodity Commission, the USDA State Block Grant Program, and the Georgia Farm Bureau, and obviously all of the members of our labs who do the hard work to get these results. With that, I will take maybe 1 question, if you have them, and I'm happy to talk afterwards to you as well.
Thank you so much to Dr. Idle for that talk. Our next speaker is Dr. Philip Harmon from the University of Florida.
Thank you very much. I appreciate the invitation to be here today to speak about an emerging disease of blueberries called algal stem blotch. And I characterize it as emerging because it's not new. It's been around for a long time. However, it has emerged within the last couple of years as a problem in South Georgia, one that we have seen in South Florida, move up through north central Florida and into our major production areas as well. It's a disease that I'd categorize as sort of an oddity. It's caused by an algal pathogen, which is not the usual. We are more accustomed to talking about fungal pathogens, of course, on blueberries, the occasional bacterium, and then a few viruses. This is an algal pathogen that we're going to talk about today, some of the interesting biology of it, and then why I would also say that this is one of those curiosity diseases that we have some good, solid management for, and that we haven't done a tremendous amount of research on because it's not a top priority for our growers. As far as recognizing this, it is a disease that moves slowly, but one that can sneak up on you. And so in the picture here, you can see some blueberries that look like they may be a little hungry. They have some yellow canes sticking up above the foliage. This is algal stem blotch, and it's sort of mid to early stages of infestation of a field. This is down in central Florida in the Haines City area. One of the first times that I saw this become a production problem, and that was around about 2007. We see it down in South Florida in our southernmost production areas ever since I started at University of Florida back in 2003. When it becomes severe, we'll see leaves that have a bleached white to yellow appearance. We'll see plants that lose their vigor. We'll see plants that do not produce as much fruit or loss in production. This could be caused by a lot of different things, but the unique sort of look to it is a white to almost white bleaching of the leaves that are attached to a single cane. And here's another picture. It's a little more white in color, and you know, all of these leaves attached to these central canes, if you follow these down, there'll be one main cane that's infected by this algal species. I'll show you pictures of that in just a second. And everything upstream of that infection has this white to bleached out look to it. This is pretty unique, but if this is all I showed you as far as the symptom of algal stem blotch, what would be the first thing to come to your mind as far as a potential cause for that? Lack of nutrients is a great guess. Yeah, lack of nutrients could be it. Where we're going to see differences here is that this is going to occur on one cane or two on the plant versus the whole plant. It's not going to be on one particular age group. If you've used some of the herbicides in the site of action 27 group, namely Callisto for blueberry, it is a pigment inhibitor. It can cause a white discoloration on some plants. And there are others that are used in other crops. So Callisto we use sometimes in blueberries, not a lot I don't think. Command, Balance Flex, Impact are used in corn and soybeans. These are herbicides that can inhibit chlorophyll production, or more specifically inhibit carotenoid production, which protects chlorophyll. So sun will bleach that chlorophyll out, turn the leaves to white to a yellow discoloration. Deficiency is another look-alike symptom that we can see. This is different because it's a disease. It doesn't go away when we correct those other things, but these are the things that we look for first when we see blueberries that are not performing well, that have a white to yellow discoloration. When we rule these out, then we look for some of the other symptoms and signs of this algal disease that we're talking about today. So why do we see this yellow and white symptom? If it's not due to herbicide, it's not due to a deficiency. First, we won't see the algal pathogen on the yellow leaves themselves. So when we look at those, we're not going to find any algae growing on them. The algal infection is going to occur on the stem, downstream vasculature from the leaves that are affected. So if we follow those canes down, at some point we will see a red blister-like lesion. We'll see the red fuzzy growth of the algal pathogen. And the reason why we see these upstream symptoms is still not clearly understood, but it's hypothesized. We know these algae can produce toxins. It's possible one of these toxins is mimicking one of the herbicides that causes this chlorophyll degradation or this pigment inhibition. And so those are hypotheses. Again, this is a very minor pathogen. It hasn't been studied a lot outside of Southeast Asia and where it affects tea and other tropical crops. It is a major issue in tropical areas on tropical fruits, tea, as I mentioned, and others. So there is a little bit of work that has been done, mostly in India, that we can draw some things from that I'll talk about here in just a minute. So it's possible these things are producing toxins. Truth is, we don't really know. We also don't know, because it's very difficult to work with, whether these pathogens, to what degree they're causing the decline of the plants, or if they're showing up on plants that are already in decline due to other reasons. Sort of like the Spanish moss problem with live oaks. They don't cause a decline in live oaks or pecan trees, but when we have declining trees, that's where we're going to see most of that bryophyte, that Spanish moss growing. So in this case, we do believe that it's causing the thinning and the lack of vigor. But again, the research hasn't been done to clearly show how such a small infection on the canes can result in such a large reduction in plant vigor. So it's possible that this can occur and does occur with other problems as well as the algal infection. So what do those infections look like on the canes down below those yellow leaves? If you follow the leaves down onto the canes, you're going to see red blotches like this in the times of year in the wintertime whenever this pathogen is not sporulating. So here we have red blisters on the cane of a blueberry plant at the end of the growing season, just as it's starting to form bark. As the bark forms, these become more difficult to see and more difficult to differentiate. It starts a year before, when the canes are small and juvenile, as little red blisters. When it becomes severe and when we have the right environmental conditions, those red blister-like lesions will produce an orange felt growth of the algal pathogen. And so that bright orange fuzzy growth is the number one sign that what we're dealing with here is algal stem blotch. This is a ton of really tiny, small sporulating structures that produce sporangiospores. So similar to fungi, these algal pathogens produce spores. These spores are moved around in the wind. They're also moved around in splashed rain. The pathogen can grow through bark. So you see the orange fuzzy growth here. Again, this is going to occur in the summer. Those yellow leaves are going to be upstream and the top of these leaves attached to this particular cane and all the smaller canes coming off of it. And this orange fuzzy growth doesn't occur for any other reason. We can see green algae where we have excess water, we have really humid conditions. This is a different kind of growth than this red orange coloration of the algal stem blotch pathogen. If we look closer at these under the microscope, you'll see these little spore heads that are produced, and then tons and tons of little sporangiospores on the tips of those are produced, again, when it's wet, when we have humidity, and when we have rain. These are blown around if they dry out, like fungal spores, but once they hit a little film of water, they germinate, and then you get a multiplier effect of 6 to 64 times, because each one of these little spores that's produced on the edge of one of these gray or orange fuzzy growths then secondly germinates into zoospores that swim in water, and it multiplies either 8 to 64 spores per each one of these that it's produced. So although this is a very slow growing pathogen, once it becomes established, it produces tremendous numbers of spores available to infect canes during the favorable conditions, which are hot, humid, tropical sorts of environments that we see in South Florida and that we increasingly are seeing in Central Florida and South Georgia. There are also differences in the varieties that we see as far as their susceptibility. Some varieties we see this as a problem much more commonly and much more severely than on others. I'll talk a little bit more about that in a couple of slides. We can do some microscopy and see that where these algal pathogens are growing is just below the bark and above the wood tissues. They're red in here because this is a fluorescence micrograph, and so these algae actually have chlorophyll in them. They can make their own food, but they're bums. They don't. They just like to live off of our blueberry plants, and so while they're growing on the blueberry plant, they don't produce the chlorophyll to make their own food. They steal it from the blueberry just under the bark and just above the xylem wood in that phloem tissue there in the blueberry stem. They steal the nutrients that are present there that the plant would use to produce fruit or grow, and then proliferate themselves in these, again, lesions on the canes. This one is on rose, so this can also affect roses, can affect brambles. This is a major problem for us in blackberries. One of the reasons we don't grow a lot of blackberries in Florida is because of this disease and others. On rose, if you incubate it long enough, it will turn green and start to grow out of the individual stems. Here in the lower right, you can see underneath the bark this blister-like lesion. That's where the algae is growing, just under the surface. We believe it's secreting toxins there, which move into the plant's vasculature and cause that bleaching out and lack of vigor and growth and production of our blueberries. And other places that we might see these particular types of related algae include on our magnolia leaves. Here on the left, you'll see algal colonies on the surface of a magnolia leaf. Here on the right, it's one of my favorite camellias in my yard right now, also affected by the algal leaf spot down here. And this is a related pathogen. Cephalurus fluorescens is the name of the pathogen. These don't cross-infect from blackberry to blueberry, we don't believe, or from magnolia to blueberry, etc. But they're in a related group that has a similar biology. When these things form lichens with fungal species, they become strigula, a different name and a different biology, makes them a little different to try to control. We do occasionally see them on the leaves of blueberry in our evergreen production systems in South Florida. It's a minor component of the disease and life cycle of the algal pathogen, and not one that contributes really to the disease on the canes. That's our main concern. So here it is on a blueberry leaf, not generally contributing a lot to the disease cycle of the cane dieback and lack of vigor. This is what we call an assistant professor killer. If you look here, this is how much it has grown in 6 years since 2018 whenever I isolated it. So we can work with these in the lab, but it takes multiple years to get them to grow just a tiny little bit out there, and they don't sporulate once we get them in culture. They stop growing red, they start making their own food and producing chlorophyll. That's why they turn green. And we can isolate it, and we're doing some work with genetics to try to determine if there are differences in the populations that we see between the magnolias, the blackberries, the arboreum, the blueberries, etc. And so that's another topic for another time as far as the research, the basic research that we're doing to try to understand better these algal pathogens. From a practical standpoint, understanding the life cycle is important, and we know that there are tons of spores produced, as I mentioned. Each zoosporangium that's produced on that little red fuzzy growth can germinate and produce 8 to 64 zoospores. If we look at those little sections of stem that show that red fuzzy growth, there was a group, Brooks et al. 2015, that actually quantified how many of those sporangia can it produce and how many zoospores that are water motile swimming spores are produced from those. And it turns out it's somewhere between 72 and 198,000 spores per centimeter squared, so per third of an inch squared. Tons and tons of spores are produced. It takes a long time for it to grow up to the point where you'll see that orange sporulation on the canes in that quantity and that severity that I showed these earlier pictures. But once it does, it becomes a very difficult problem to get ahead of, to get on top of, with the management options that I'm going to talk about next. So prevention, like many of our diseases, is even more important for this disease. We can kill it. It is sensitive to copper and other pesticides that we can use, but we have to get out ahead. Again, stressed plants are more susceptible, and the disease cycle can occur a little bit more quickly. So as far as managing this disease in blueberry, what we know that has worked has been multiple repeat applications of copper and potentially some other fungicides that are used in blackberry as well. But where we're pulling this data is not necessarily from trial work that we've done. We're pulling from areas of research on other crops with related organisms. So there's some chemical control data from India on tea leaves that are major commodity there and grown are also susceptible to a related Cephalurus fluorescens. They have used fungicides, carbendazim and hexaconazole. This is a DMI fungicide related to propiconazole and some of the other newer products that we have. So we're looking at those as potentials, although where they have been used regularly to control leaf diseases, we haven't seen a tremendous amount of algal stem blotch suppression from them. Carbendazim is used in India. It's related to thiophanate methyl, which is not registered for blueberry here in the States. We haven't seen or heard where off-label use has occurred a lot of control there either. So these are options that we're considering, but not likely to be silver bullet cures, certainly for this particular problem. They've also looked at biological controls, including streptomycin species. We have some of those in the States here. Actinovate is one that has shown some promise outside, something that we're interested in looking at further moving forward. If we look at other crops closer to home, of course, Dr. Brennan, Dr. Sherm at the University of Georgia just recently published some work that they had conducted on blackberry. Blackberry is susceptible to orange cane blotch. That's a similar disease. Blackberry, of course, a very different crop with a biennial system of primicanes, floricanes. This life cycle is much different than our perennially grown blueberries. What they found and published is that only potassium phosphite in the Prophyte product was consistently reducing disease for blackberries. And in their paper, they talk about some of the limitations, of course, with the field research and with how slow this thing grows, in that they used products on about a 4+ week interval, and a little later in the season then potentially this pathogen starts to sporulate. So we also know in blueberry, if we don't put these out at least monthly, and more likely every 2 weeks, we're not going to get on top of this or prevent it. And we also know that where we have the applications go out too late into the midsummer, we're going to have sporulation that's already occurred, infections, and once those infections occur, coppers are not going to get into the plant at all. The phosphite products can, but they have limited efficacy on this particular disease. So we need additional research here. We do know copper fungicides can help from anecdotal reports from growers and from observational trials that we've done. And where we have high disease pressure, we need to split up our applications of copper hydroxide into every 14 days, is what we've found to be most efficient, by going with a lower rate and a more frequent application through the summer period. When we see that orange sporulation is when we've seen the best prevention of additional infection and additional development of disease. Phytes may help, and they're good to use for some leaf spots and for Phytophthora management, so we want to go ahead and use those as well, but copper products have been most promising. So for an example, Cocide 3000, we have between 1 and 3.5 pounds per acre that we can use on a 7 to 28 day scale. Once we get the problem under control, if we're strictly preventive, we can go with the high rate and go with the longest interval. If we're dealing with a situation we know the disease is present, I'd recommend going to a 14 day or maybe a 21 day, and then go with a mid rate somewhere in the 2 pounds per acre range. We want to follow the instructions on that Cocide 3000 label for bacterial canker or Phomopsis twig blight, or whatever disease the particular product that you're working with has for blueberry. It has to be labeled for blueberry. It doesn't have to be labeled for the particular disease, algal stem blotch, to legally use that. Magnabond, Badge, and then homemade Bordeaux mixture have also been reported to have efficacy, and there are other options there that will get product copper out into the field for us. Coppers are good not just for this algal stem blotch, but leaf spots and others as well. So they can be incorporated in a fungicide management program for summer disease leaf maintenance. Sprayers do need to achieve good cane coverage to apply that protective coating on the canes before infection occurs. Overhead irrigation is a killer for us in this disease. Where we're using it, we're going to have major issues and almost impossible to overcome when we're growing a susceptible variety like farthing. Some of these others here can get it as well. In particular with Jewel and Emerald, we find we don't see that disease until it becomes a 7 to 8 year old planting, at which time those will also become susceptible. Arboreum rootstock is an area of research that Dr. Williamson and others at UF have been working on. It unfortunately is very susceptible to algal stem blotch as well. We did some work with curative treatments, looking at trying to shut down sporulation with herbicides that are labeled for in different cases and different uses for algae suppression, either through ponds or through other uses. We made applications in summer, and then we collected those canes that already had the disease to look to see if our herbicides could prevent sporulation from occurring as a curative type option. We looked at these herbicides, diuron, hexazione, mesotrione, these others. Still would like to look at simazine. Bottom line is they didn't work, and so they're also off-label, illegal to use this way. So don't use these. There was no significant reduction of algal stem blotch from this. We also looked at Time Guard and organic oil as a potential method for inhibiting spore production, where we already knew we had a problem in chickadee. Looked at severely pruned and lightly pruned plants on a cooperator farm, and then measured the quantity of sporulation produced on these after multiple applications of this time-based oil, and it did not work either for us in these particular trials. So it's something that we're continuing to look at. We do have good preventive applications and efficacy where we use our copper products, and so rely on those, and Phytes may help us out a little bit as well. Appreciate your time for the invitation to come speak today. Thank you very much.
Thank you, Dr. Harmon, for your time and expertise. Our next speaker is Dr. Angelos Delsidis from the University of Georgia talking about post-harvest in blueberry.
Thank you. Hey, everyone. I'll be talking to you about some work we've done this past year on some of the quality issues of blueberry we see in Georgia and some general handling guidelines for blueberry. As you all know, blueberry is a very important crop both for the US and for Georgia. We are the third leading state in production and the highest in reported acres. And we're growing two main types of blueberries. The southern high bush is about 60% of our acreage, and then 40% is our rabbiteye. The gate value, the farm gate value is about $348 million, with the productivity of about 4,500 pounds per acre, which is lower than other states in the Northwest. The post-harvest quality and acceptability they're linked, and they're mainly based on the quality and the texture. And we have many factors. Why is this advancing on its own? Sorry. The pre-harvest factors, such as the environmental factors, the quality of the soil, the cultural practices, all affect the post-harvest quality. Also, the harvesting mechanisms, I apologize, this is advancing for no reason. The harvesting mechanisms and handling, processing, and storage conditions affect the quality as well. So in the past few years, we've been seeing issues with fruit quality that's been reported by the growers. We have some fruit split, oozing from the stem end, as well as internal browning. And you can see here pictures of fruit that have this issue. The internal browning, initially we thought it could be potentially due to freeze damage. The splitting and oozing could be due to irregularity in harvesting schedule. And that over-ripening in general leads to weakened celled walls. So the objective of this work was to evaluate the effects of various picking intervals on the storability and quality of Georgia-grown blueberries. So what we did is we had two locations, and we had two cultivars, a southern highbush, meadowlark, and a rabbiteye, which is Bridewell. And we had two harvests, 7 days apart. Sorry about that. The harvest one was done on May 17 for the southern highbush and June 2nd for the rabbiteye, and the second harvest was done a week later. We did 5 replications for this study. We stored the fruit at 1 degree C or 34 Fahrenheit and 85% relative humidity, and the fruit quality assessment was done weekly for up to 3 weeks of storage. We did visual assessment of the fruit. We calculated moisture loss and dry weight, color changes, total soluble solid concentration, titratable acidity, as well as anthocyanin content. Here are pictures of the Bridewell fruit, and I'm going to show you some data. The firmness of Bridewell blueberries declined the longer the fruit remained on the bush, both at harvest as well as after storage. You can see the deep blue is the harvest, the first harvest, and the second one, the lighter blue, is the second harvest, 7 days later. The fruit size was larger, especially during storage in harvest 2. Whereas it declined more rapidly during storage for harvest 1. Total soluble solids, we had initially higher total soluble solids for harvest 1 for Bridewell, and then during storage they were pretty close. The acidity was always higher for the first storage, and it declined over time. The firmness of meadowlark cultivar initially there was no difference, whereas during storage we had lower firmness for the delayed harvest, for harvest 2, as you can see here. The fruit size in this cultivar, we had larger fruit size, the longer the berry remained on the plant as expected. And then we had a smaller size in harvest 1, which declined over time. Soluble solids for meadowlark, initially there was no change, but we had higher soluble solid concentration for the harvest 2. So in conclusion, from this preliminary study we did this past year, we found that fruit from harvest 2 had lower firmness values after 7, 14, and 21 days of storage. The moisture loss was about 3% every 7 days of storage. We found that the delayed harvest reduced fruit storability by negatively contributing to fruit firmness and to other fruit quality characteristics. In general, we saw that there was a breakdown of starches into sugars as expected, and the acids were metabolized by enzymes, and there was a degradation of monomeric anthocyanins. We recommend that we have timely harvests of intervals less than 7 days apart to maintain fruit quality and storage potential. Here are some suggestions for berry pickers. We need to make sure they have clean hands. They pick all the berries, the ripe berries on the bush before moving on, and they harvest only well-ripened fruit. They should leave the immature fruit. I so apologize about that. I think I want to exit it. And leave the unripened berry on the fruit for the next harvest. Avoid overfilling their hands and the buckets. Leave the trash behind, and never allow this fruit on the sun. With that, I'd like to mention that machine harvesting, which is the norm right now, we have a lot of issues to quality because of the height of the fall that is causing bruising to the berries. This bruising shortens the shelf life. In general, there's varietal differences thanks to the stem scars, and we need to make sure the machines are staying clean, and there's debris removal every so often to avoid, I'm gonna do it this way, I'm sorry guys, to avoid issues with diseases being transferred from one fruit to the other. I was looking for a recent study on the quality of the berries after storage in the store, and I found a study from Spain that was published late last year in 2023, and it investigated how fruit arrives in the outlets before purchase by the consumer. They looked into the firmness, the physical chemical parameters, phenolic compounds, and fungal growth. They saw that the fruit arrives with no fungal decay at the time of purchase. And what was interesting to me that they found very low values of total phenolics and anthocyanins, much lower than the ones we see here in the US. They saw also no differences between conventionally and organically produced blueberries. And a common characteristic of the samples analyzed was that they had low maturity index values. So they do something similar that we do. Often they harvest very early to make sure the fruit is firm enough and makes it to the market, but they don't focus as much to the sweetness and the other qualities that consumers like. With that, I apologize about this issue. I'd like to thank Amit Kodara, who did most of the work, as well as my team and Delfina Rubio's lab, who helped with this study. Thank you. I don't know why it was advancing on its own. I think we have time for questions.
Yeah, you have time for questions. We do have time for questions if you have any question for Dr. Delsidis. Our next speaker is Dr. Jinru Chen. She's also from the University of Georgia. She's going to be talking about the cleaning and sanitation for blueberry machine harvesters.
Good afternoon, everyone. Thank you very much for sticking around in the afternoon of last conference day. What I'm going to talk about part of the results of a 2-year research project that just ended about a year ago. The title of that project is evaluating food safety challenges of blueberry harvesting. cleaning and sanitation of machine harvester is only part of that project. This slide has the members of the project team. Beside myself, I have Dr. Wei-Qiang Yang, professor from Oregon State University, as one of my co-PIs. He is a state-funded blueberry extension specialist in the area. When the pre-proposal put together, it was a collaboration between myself and Dr. Eric Smith before he left the university. After he left, Dr. Renee Holland, who graciously stepped in and worked with me till the end of the project. I also have a couple of collaborators, including Dr. Fumi Takeda from USDARS, and also a few professors from either Rutgers University or Michigan State University. The call for research proposal came in 2020, in the middle of the pandemic. At that time, Center for Produce Safety, a trade organization out of California, identified the research need for the blueberry industry. They think like a researcher like myself should address food safety issues related to blueberry harvesting, including the hygiene conditions of blueberry harvesters and also harvesting equipment. So based on that, we put up together a proposal with 3 different objectives. The first one, we want to do an online survey to collect information about the cleaning and sanitation protocols that growers or packers are using to clean their harvest containers and machine harvesters. The second objective is a laboratory study. I'm not going to spend too much time on that. The third one, part of it is today the focus of my presentation, is a field validation study. We basically validated the efficacy of a few cleaning sanitation approaches used by growers and packers to clean their hand picking bucket, like berry logs or flat, and also machine harvesters. This part of the study is very important because based on the results of our online survey, only about 75% of the growers sanitize their harvest containers routinely. About 3% of the growers never did. Similarly, about 2% of the growers never sanitize their harvest, the machine harvesters. So in this harvest survey, we sampled a total of 10 harvesters. These include 4 harvesters from 2 facilities in Georgia, and can't make this work, 6 machine harvesters from 2 facilities in Oregon. Each of these harvesters was sampled twice in a single harvest season. All these 10 harvesters are conventional over-the-row machine harvesters without any modification done on them. In Georgia, among the 4 harvesters, 2 were bottom loaders, another 2 top loaders. All the 6 harvesters in Oregon are top loaders. The sampling scheme we used, we followed the sampling scheme Dr. Renee Holland and Dr. Harold Sherm did a few years back, as shown in this photo. So we swabbed the upper and lower portion of the sidewall, and also upper and the lower portion of the beating bar. And the fish scale, like this one fish scale, the horizontal conveyor, berry log. In Oregon, they use berry flat instead of lugs. And the vertical conveyor that transport the fruits from the bottom platform to the top platform, and also the flap on the top form of the machine. We sample two different surfaces. One surface is cleaned surface right after cleaning and sanitation. Another type of surface is just used but not yet cleaned and sanitized surfaces. We located in Griffin, Georgia. Alma is 3.5 hours away from us. Basically, students drive to Alma in late afternoon. After 7 to 9 hours of harvesting, we swab the dirty surface or use not yet clean surface. And then after that, growers do whatever they need to do in terms of cleaning and sanitation. And then they leave the surface dry, and then next morning, first thing we come in is to swab the cleaned surface. As you can see in the harvester, there are surfaces with irregular shapes. For flat surface, we swab 100 square centimeter area. For the area with the irregular shape, we make sure the swabbed area is clearly is precisely measured. We calculate the precise area we swab for the results. So this is the results coming out from the Georgia Harvest Survey. I try not to compare the two states and put the results together. I want to bring your attention to the first group of two bars here. As you can, one means the first facility in Georgia, and two means the second facility in Georgia. As you can see from this slide, the value that come out from the second facility is relatively lower than the first one. So this bar represents the value or levels of a group of microorganisms called total aerobic counts. This is not anything like a pathogens or anything. This is a group of organisms that can grow under regular atmosphere at 37 degrees centigrade. There's no regulatory standards about how high or how low this count should be. But generally, people believe a freshly cleaned surface should have a total aerobic count lower than 2 log CFU per square centimeter, or 100 cells per square centimeter. So go back to this slide, we can see in this particular situation, it seemed like a facility 2 did a slightly better job than facility 1. The table to your right shows what they did with the cleaning and sanitation. For facility 1, they clean harvesters daily. They use a hand to remove debris, and then use a paper towel to remove other residues. And then they use 1.5% ppm chlorine for sanitation, followed by rinsing and drying and so on. For facility 2, they clean sanitized harvesters every 2-3 days, but they have a deep cleaning weekly. It seems they have a better pre-sanitation cleaning step. They use a blower to remove debris, and then use a cloth soaked with soapy water to clean the soil and dirt before using 200 ppm quaternary ammonia for sanitation. And now I want to bring your attention to these two bars. These two bars shows the hygiene condition of the surfaces. One surface I said is like a cleaned surface. This is right after cleaning and sanitation. And this is just I don't want to use the word dirty surface. It's used, but not yet cleaned surfaces. And these two bars shows basically sanitation really worked, because after sanitation, the counts on the cleaning surface is a lot lower than the used surface. Other than that, we identified a few hot spots on the harvester. This is basically concentrated on the bottom of the machine, lower portion of the machine. You know, as I show here, the three bars with the red lettering. And these are the size called EFH. I have these listed here. I think the highest one is H. That's a vertical conveyor, you know, that transfer fruits from lower portion of the machine to the top portion of the machine. And understandably, a fish scale, the fruit catching place, you know, one scale overlap the other, that sometimes hold more bacterial cells. So basically, the fish scale and two different conveyors, these are the hot spots. So these are the area perhaps we should pay a little more attention when it comes to cleaning and sanitation. Another interesting thing I want to point out are these two. Because you know, these we sampled these 10 harvesters twice in a single harvest season. So we have two different visits. So we have done this in a while, not only with the blueberry, we also work with the peach industry and different things. So we find out it's always true, like you know, the results come out from the second visit is lower than the first one. I kind of take this as a positive results, because when they go in, growers and packers know, you know, what we're looking for. They make a better effort the second time around. Apparently, you know, the results is a whole lot better. And this figure shows the similar results, but from the harvester out of Oregon. They also have two different facilities. The second facility also did a better job than the first one. It's very interesting. I think when we did this work, their state-paid extension agent play a lot of role. He worked with both facilities. These two facilities, they have the similar cleaning protocols. As you can see, they clean, sanitize daily. And then they use blowers to remove debris, and use high pressure water to clean them first. But the sanitizer they use are different. The first facility use 200 ppm chlorine. The second one use 80 ppm peracetic acid. After that, their procedures are pretty similar too. They use high pressure water to rinse it off, and followed by air dry. And very interesting, I think their hot spots is pretty much the same as what we found in Georgia. So that pretty much confirmed, you know, these are the area we should pay more attention. Okay. So from what we did in this study, we can pretty much see growers use different approaches to clean, sanitize harvesters. These protocols more or less are working. They have different efficacies. The study did have limitations because we only work with 4 different facilities in 2 different states. Although it's over 300 samples, but if we do more facility in more states, we'll probably get much bigger picture. But even with this limited study, we can see a few points. The first one is sanitation did make a difference. The clean surface, did have a lower microbial counts than the used surface. Another one, I think the pre-sanitation cleaning steps is very important. In order to get a good sanitation results, the surface should be thoroughly cleaned before sanitation happens. One other thing we noticed, not necessarily from a harvest study, but from hand-picking buckets and lugs and flasks, whenever there is a heavy buildup on the surface of the harvester, sometimes a little scrubbing is necessary for thoroughly clean the surface. I understand, growers may have a different approach than from what we presented here. But no matter what a protocol you use, just make sure the designed protocol is followed, and then the efficacy is validated to make sure it works well. This last slide, I have a few acknowledgments. The project was funded by Center for Produce Safety and also California Department of Food and Agriculture. I'm from Georgia. I think this California Department of Food and Agriculture, they provide a matching fund for the Center for Produce Safety's grant. I appreciate all the co-PIs and collaborators for working with me. I don't really have a very good luck, and after this project, a few retired and others left university. So, but I really appreciate that they took an initiative working with me. My PhD student, Ya-Si Dai, on the picture, and Sarah Dong who is a research technician from Oregon State University. They did a very good job in collecting samples. I'm very grateful for the 2 growers in Georgia, the 2 growers in Oregon for working with us. We are a lot of trouble sometimes in your a very busy harvest season. We kind of in the bay and try to collect samples. But without this participation, we won't be able to collect any sample or for any suggestions or advice we can provide. Anyway, thank you very much for your attention.
Thank you so much, Dr. Chen, for your time and expertise. Our next speaker, we are going to welcome back to Dr. Rachel Idle.
Well, thank you for sticking around for the last talk of the blueberry session. Hope it will be interesting and worth the wait. So today I'd like to present some results that we had on some fruit cracking preventative strategies that we worked on in my lab, and then also to see their effect on the resulting fruit quality of treatments. Okay, so I think everybody who was here earlier is still the same, so I won't mention this. Obviously, blueberry is big in our state. A big problem for both types of southern highbush and rabbit-eye blueberries is fruit cracking during our harvest interval. So why does cracking occur? First of all, it occurs during fruit ripening, and it's believed to be after periods of precipitation changes. So basically you have really dry conditions followed by very heavy rainfall. You can actually create cracking if you do this. If you have very dry and then you hit your plants really hard with water, you can kind of create this, this problem yourself. The basic premise is the mesocarp, or basically that flesh inside the skin, starts to expand with water uptake, and it expands more easily and faster than the skin can keep up with. And basically, it just cracks the skin and it causes it to split, kind of like a stretch mark on human skin. If your skin is, your fat underneath your skin is growing faster than your skin can keep up with, you're going to get a stretch mark. To put it very simply, that's kind of what's happening with blueberry fruit. One thing that's exacerbating this are increasing temperatures, basically changing climate patterns in the world. So basically more weather fluctuations. So we have increased periods of drought that we're having to irrigate, or then we have really wet weeks after that, and the temperature changes as well are not helping it either. And this is very important because any amount of fruit cracking will render the fruit unmarketable. It's not like perhaps in other crops where maybe it's a blemish and it might then be keyed to a processing market. Blueberries are expected to just be this beautiful, perfect blueberry, so any sort of little bird pick or crack or anything makes it not desirable for selling. The main current prevention strategy is cultivar selection. However, there's not been a lot of research on fruit cracking. The most recent that I was able to find was back in 2006 out of Mississippi, some of the rabbit eye work showing that Premier had less splitting than Tiff Blue. So breeders are currently working to breed resistance of cracking into new cultivars, but that, as you know, it takes time. So what can we do in the meantime to help with cracking that won't just involve planting new cultivars? There's retractable row covers that we can do to kind of exclude some of the rainfall events as they come, soil moisture prevention, foliar sprays, cuticle protectants, and even wind machines or helicopters to basically try to dry that out so it doesn't get too wet. So my program, we did some trial work with Cultiva looking at parka as cracking prevention. And what is parka? It's basically like, I think of it like a little raincoat for the blueberry. It's a cuticle protectant spray, so it's a spray that you spray on your plant, on the fruit, and it stretches with the fruit skin, so as it grows and shrinks, so does the compound. And it basically, like a raincoat, repels the moisture away from the fruit skin and is supposed to increase that cuticle layer, or the basically the outside of the blueberry, and prevents the water from entering it. In some trials that Cultiva had done, this does not work with my program, but previous to work with me, it's been shown to reduce rain-induced cracking by up to 40%. But we haven't shown, or it hasn't been examined, what happens to the fruit quality. So anything you're spraying on the fruit, you wonder, well, what's happening to it? Are you making it taste better, worse? Is the texture going down the toilet? Is it becoming more mealy, and so forth? So we wanted to examine that. So first of all, what is fruit quality? Why should we care about it? Put very simply, it's the combination of physical and chemical attributes that determine the quality of the fruit. You can think of it basically as four main areas. So first, the appearance. When you go as a consumer into the grocery store, you're looking at something big and beautiful and shiny. Appearance is probably the number one. After appearance, texture. When you pop something into your mouth, you don't want it to have some sort of weird texture. And then if you have a good texture, you're going to wait to have that beautiful taste of the flavor and the sweetness and the sweet-tart balance, so texture and taste. And then health attributes such as antioxidants. Arguably, I would say the two main areas outside of appearance are texture and taste. There was a breeder, he's since retired, Dr. John Clark out of University of Arkansas. I remember mentioning in one of his talks is that and I think he was talking about this in relation to muscadine, but basically if the texture isn't good, they're not going to keep it in their mouth long enough to see what it tastes like. So I would argue as well that texture is probably our main attribute to look at in blueberry for quality. For blueberry, that mainly is looking at the fruit skin toughness or strength, and then the fruit overall firmness. And then the second main category, the taste or flavor, so not looking at all the little flavor volatiles, but just very simply the sweetness and the acidity of that fruit. And it's really important to consider post-harvest anything that we're spraying on the fruit that we're about to harvest, how is it affecting those quality traits? So what do we know so far about Parka on perennial fruit quality? Well, it's been published in 3 different perennial fruit crops. The first is juju fruit, where Parka and some jib treatments were done. They saw no negative effects on quality, and they actually saw an increase in sugar content and acidity. In sweet cherry, there were 2 studies done. One showed an increase again in sweetness, no increase in sweetness, but an increase in acidity, and another one showed no change. And clementine, where they saw an increase in crop yield, and this was probably most likely due to a decrease in the fruit drop that was happening. So what do we know about Parka on blueberry quality? Short answer, not much. There's only been 1 published study to date on blueberry, and it was in 2022. It's 1 blueberry cultivar, I believe. It's blue crop, so northern highbush variety, and it did show a slight decrease in fruit size and an increase in the color characteristics. But again, only 1 variety, so we don't have any information on any of the rest of the northern highbush and no information on any southern or rabbit eye varieties. So what was our main objective? Well, we wanted to evaluate it for cracking as well. I forgot to put that on the slide. But in addition to the cracking ability, what does it do to the fruit quality of the fruit after it's been applied? So we did this for 2 seasons. We basically had 3 treatments that we did across both years. So we had, and we just followed the label recommendations of the product. So that's why I don't have the spray information or the application rates here. We sprayed at blue fruit, or I guess chronologically green fruit, then blue fruit, green and blue fruit, and we did that across 2 years. And we did it on powder blue both years. In the second year, we also added it Brightwell and an organic formulation sprayed at the blue fruit stage. This was also done at a commercial farm in Alma in a randomized complete block design. So we had 3 treatments, or I mentioned the treatments already. We had 5 reps, and we had 2 collection dates. These were approximately when about 50% of the fruit was ripe. So we tried not to get when the fruit was underripe or overripe hanging on the plant, tried to get that optimal midpoint for that. And then we took them back to the lab to look at their quality characteristics. The fruit cracking assay was done in collaboration with Dr. Renee Holland with her research and consulting company. She collected the fruit the day after harvest, brought the fruit to room temperature, and basically submerged the fruit in water overnight, and in the morning scored them. And it was a quantitative score, so here you can see she counted the number of cracks. So this is an example of 1, where this is an example of a score of 3. In my lab, we did textural or size and texture. So first we went ahead and we weighed the berry, took some measurements of diameter and height, and then we did our classic textural test, the puncture probe here, that equates if you were to take a blueberry and put it between your front teeth and bite down that pop. That's a measure of the skin strength or skin toughness. And then for fruit firmness, if you were to take basically a handful of berries and put them in the cage and squish them down, it's equivalent if you were to take a handful of berries, put them in your back molars and chomp down, and you feel the fruit firmness. The remaining fruit, we went ahead and froze it for sugar and acid analysis. So we processed the juice. We first blended up everything, all the berries. Then we went ahead and poured it into tubes, and we wanted to separate it out, so we spun it really fast, and separated out the juice, separated from the pulp and the skins and the seed. And then we went ahead and collected and filtered that juice, so we had nice purified juice to go ahead and take measurements of acidity and sugar measurements on it. These are standard post-harvest measurements, so we went ahead and measured the amount of acidity and the amount of sweetness as indicated by the soluble solid content or Brix content of the fruit. And And for fruit cracking and for fruit quality, our basic premise here is we wanted to know, for fruit cracking, are our treatments different than the control? And for fruit quality, are our treatments different than the control? And if so, are they better or worse than the control? So let's take a look at the fruit cracking results for powder blue. Across both years, the treatments did not show any significant difference from the treated fruit for cracking to the untreated fruit. Here on the left are the untreated fruit, and you can see treatments of blue fruit, green fruit, and blue and green fruit combined. And then the same order over here, but also the addition of the organic formulation. We did see, again, these were not significantly different, but again, a trend that this was the lowest, the blue and green fruit stage were the lowest means as compared to the other treatments. When we went ahead and combined the data across both years, we did see a significant difference for the blue and green fruit applications in terms of cracking, and that suggests that perhaps applying Parca throughout the fruit development period for green and blue fruit shows a reduction in cracking. But we're seeing trends here when the years are separate and then a significance here when they're combined. So this suggests to me that if we increase the fruit sample size, evaluation of additional years or cultivars, then we may be able to understand what's happening a little bit clearer. In Brightwell, no significant difference between the untreated fruit and the treated fruit for level of cracking. But again, we did see a trend with the green and blue fruit application having a lower mean. And our basic result from this is it suggests that perhaps the blue and green fruit application or applying Parca through fruit development can reduce cracking, but we need to evaluate that further to give that recommendation. Okay, so going through the physical traits for powder blue, we looked at this for 2 years and we evaluated fruit size characteristics, so berry weight, berry diameter across the equator, berry height, how tall is the berry, 2 measurements of skin strength, and a measurement of fruit firmness. And I'll go through these generally and give the summary of it clearer at the end. So for berry weight for year 1, we saw 2 treatments had larger fruit than the untreated control. In the second year, all treatments were larger than the untreated control. For berry diameter, or heavier rather, for berry diameter, one treatment, the blue and green fruit stage, was larger than the untreated control. And again, I'm comparing this to the treated control because I don't really care so much about how the treatments compare to one another. It's more, do the treatments improve or reduce the quality compared to the status quo of the untreated fruit. So in the second year of the berry diameter, all of the treatments were larger than the untreated control. For the height, we saw similar results there as well, where we had the blue and green fruit was larger than the untreated control, and 3 out of 4 treatments were, in the second year, it was higher in 2 treatments, or I'm saying it wrong, in 3 treatments, apologize. Fruit skin strength, we did not see any change between treated fruit and the untreated fruit in the first year, and in the second year it was actually lower in some treated fruit. Fruit firmness, we saw a decrease in one treatment and an increase in another, and no change in the second year. So just to kind of summarize all of that quickly, in the first year, we suggested basically that Parca may improve the berry size, so it may make berries larger, and it may increase firmness when applied through the ripening stages, but it also may decrease fruit strength or skin strength. Looking at the chemical trace of acidity and sugar content, for acidity, it did not affect the acidity at all across both years when comparing to the control group. For the sugar content, we did see an increase in sugar content for fruit sprayed at the green and blue fruit stage in year 1, and then in year 2, we did see 2 treatments also increase in sugar content, however, not the same treatments. Sugar acid ratio, so the measure of that sweet tart balance that those of us in the fruit industry often think about. In 2021, all of the treatments were higher in that sweet tart balance than the untreated control. And in the second year, we had 2 treatments of that. So to quickly kind of summarize that in general, that Parca is not affecting the acidity content of the fruit, but it does suggest that there would be increases in the sugar content and increase in the sugar acid ratio as well. For the second year, so we evaluated Brightwell only in 1 year, the second year. In comparing the physical traits for berry size, we also saw increases in blue fruit stages applications for berry weight, increases and diameter. And for berry height, we also saw 3 out of 4 treatments increase in size as well compared to the untreated control. For the fruit skin strength, again, nothing really to write home about for this. No differences compared to the untreated control, so Parca is not really affecting that so much in Brightwell. And fruit firmness, 2 treatments were showing higher or firmer fruit than Parca. So just a general summary here that Parca may increase berry size and it may increase fruit firmness as well when sprayed throughout fruit development. And the last results for Brightwell, on looking at the acidity and sugar content. In acidity, it did jump around in comparison to what we saw with powder blue. We actually saw that the treated groups were more acidic than the untreated control, except for one treatment rather. For bricks, we saw that 2 of the treatments had higher sugar content than the untreated control, and 1 had lower. And for the sugar acid ratio, we had 1 higher and 2 lower. So this needs a little more evaluation, but a general summary from what we saw here is that Parca may increase the acid content in Brightwell, and it may increase the sugar content when sprayed at a green fruit stage. So general summary points for what we've observed so far. So for the cracking of Parca, the applications that are occurring during both green and blue fruit stages are showing the most promise in reduction of fruit cracking. However, more studies are needed to further understand this, looking at increase of cultivars or increasing the sample size to really see the trend that's occurring here. For the fruit quality, it appears Parca does not have any negative effect on the fruit quality, which is always a great thing. And on the plus side of things, it may improve some traits. So some suggestions that it improves fruit size, firmness, and sweetness, again, when applied throughout fruit development, so green and blue fruit stages. And we are hypothesizing that perhaps whenever we're spraying that on the plant, it's protecting the fruit in some way, so it helps to maintain those processes that make those taste components and the firmness and so forth, so it's protecting the fruit and maybe helping it to be of a higher quality. Again, further trials to understand this, but if I were trying to avoid cracking and really had a problem with it, I would say go ahead and give it a try at the green and blue fruit stages as per the label recommendations in your crop. There's no negative effect to it. It seems like it could provide some protection and could increase some of your fruit quality characteristics with the current results. So with that, I'd like to thank Cultiva for the funding for the trial, and again, all the members of my program and Dario's Peach program for their help in conducting the research. And with that, I'll take any questions that you may have. Yes?
So when it comes to looking at cracking,
Mm-hmm.
the majority, if not all, the moisture comes to the skin of the berry, not up to the plant?
That is, that's basically the premise of what the thought is. I have seen where, of course, our roots are what's taking up the water into the plant, but it seems like what is really affecting it is the water on the fruit itself. Because they can uptake water, not the same way that the root systems do to feed the whole plant, but it's the water on the fruit. And if you think about rabbit-eye too, compared to like southern highbush, rabbit-eye can hang on it can shrivel and shrink and expand as it goes with irrigation, whereas southern highbush depending upon the variety, of course, but I feel like it shatters a lot more easily if you have a heavy thunderstorm. But it's just that kind of shrink and expansion that causes it. Okay.
Thank you so much, Dr. Idle. Well, this was the last speaker, and for the attendees, thank you so much for being in this session. Please remember that we have some surveys for the growers and some surveys for qualify if you want to put a grade into this session. If you have any concern, please don't forget to do the surveys. You can take the surveys with you and give it back maybe to your technician back in the farm. And also we have the QR codes that Dr. Rubio showed in her presentation.